Microbial contamination is costly to companies in terms of equipment damage, production downtime, product contamination investigation, and energy losses. Now, companies seek to understand the sources of contamination, environmental condition, facility, and equipment design that can lead to microbial colonization and proliferation. In addition, companies have started to evaluate alternate strategies for sanitation and cleaning. Contamination is the introduction of impurities of a chemical or microbiological nature or a foreign matter into or onto a raw material intermediate or API, for example occurring during production, sampling, packing or repacking, storing, and transporting. Most microorganisms found in pharmaceutical manufacturing environment are gram-positive bacilli and gram-positive cocci, generally human purine types, yeast, Flaminus fungi. Many of these types are non-pathogenic and therefore not considered objectionable. Generally, contamination sources are equipment, process, material, utilizers, personnel, and facility. Most raw materials for pharmaceutical products support some forms of microbial growth, depending on the nutritive properties and moisture content. Hence, dry powder or tablet are capable of undergoing some form of microbial spoilage and degradation. The more serious problem of microbial contamination of tablet is there are no obvious signs of spoilage. The surveillance of microorganisms in particular environment is something influenced by the presence of relatively inert materials. Thus, microbes can be more resistant to heat and dissociation in presence of starch, cassia, and gelatin. Personal hygiene is also could be source of contamination, as they could transfer contaminants from outside to inside, and also when they are not using good gloves, suits, and masks, they also could transfer contaminants. Environmental parameters including temperature, relative humidity, differential air pressures, air turbulence, and status of high efficiency particular air filter may also have a significant impact on microbial access into pharmaceutical products, as it may carry dust that carry microbes from outside to inside with traces of some harmful materials such as sulfur and lead. Equipment used in manufacture should be regularly cleaned, as if it kept without cleaning, it will transfer contaminant from product of one batch to another. And also type of process is the source of contaminant, depending on if it's open or closed process. Machines used in pharmaceutical factories is also source of contamination, as if it carries certain contaminant it will transfer it to all samples, resulting in manufacturing spoiled products. Stabilization and sterilization of pharmaceutical products should be regularly checked, because as usual pharmaceutical products contain water with organic and inorganic ingredients that are very liable for contamination. For the final product, the total aerobic microbial count should be under 10 power 3 CFU per gram, and the total yeast mold count should not exceed 10 power 2 CFU per gram within the finished products of oral non-aqueous preparation. Likewise, the finished products of the oral aqueous preparation should not go over the limit of 10 power 2 CFU per milliliter for total aerobic microbial count and 10 CFU milliliter for total yeast microbial count. E. coli must be absent from both categories of oral preparations. Some microorganisms show their pathogenic effect only under certain conditions of light, humidity, and temperature. Thus, shelf time period could lead to appearance of contamination if these conditions are not adjusted well.
Cross-contamination among production line is an important issue in drug manufacturing because it can put both patient and workers at risk of adverse health effects. The main sources of cross-contamination in drug manufacturing are human being, air equipments, water, and raw materials. Human beings carry normal flora such as Staphylococcus aureus and may serve as the main source of microbial cross-contamination. during manufacturing of pharmaceuticals. Production lines for a range of different active pharmaceutical ingredients are often run in parallel. It increases the risk of cross-contamination, where active ingredients from one line can be carried across to the other line through the air on workers' clothing or via contaminated equipment. The complication of cross-contamination, such as if certain sensitizing compounds such as penicillin and beta-lactam antibiotics make their way into drug production, they can trigger allergic reaction even at low levels. The risk range from inconvenient to dangerous immune response, including full-blown anaphylactic reaction, which may be fatal. There are rules and guidelines of good manufacturing practice (GMP) to prevent cross-contamination and in order to protect patients and workers as these directives continuously evolve. Manufacturing technologies change and improve as drugs become more specialized or potent. The recommendation differ from country to country. The broad trend has been to segregate the manufacturing of specific types of product. The FDA has been moving towards keeping the manufacturing of penicillin and non-penicillin based drugs separate over the last 40 years. This guideline serves to set out good practices applicable to facilities handling pharmaceutical products that contain hazardous substances such as certain hormones. This guideline primary focus is on the air conditioning and ventilation systems of the facility. Facility layout. The premises should be designed and constructed to prevent the ingress or egress of contaminants. Air Handling Unit AHU to provide the required air movement within a facility. And continue to facility layout, airlock, an enclosed space with two or more doors, which is interposed between two or more rooms. Alert limit. The alert limit is reached when the normal operating range of a critical parameter has been exceeded. Barrier technology, a system designed to segregate people from the product.